Well, good evening and welcome. I'm Mike Botchin, Dean of Biological Sciences at UC Berkeley. And together with Francis Hellman, the Dean of the Division of Mathematical and Physical Sciences, I'm delighted to be here this evening to kick this session off as part of the virtual series of science talks celebrating basic science at Berkeley. It's been a great day for us today at Berkeley with a colleague winning the Nobel Prize, but more about that from one of our speakers and from Francis Hellman at the end. I, as do most of us extroverts, really enjoy meeting people in person, but the silver lining to these virtual events is that we can reach more alumni and friends who moved away from the Bay Area. As you know, in many ways, Berkeley's history can be told as the story of paradigm-shifting discoveries. While we've always been a leader in imaging, tonight's theme, we find ourselves right now at the edge of some truly groundbreaking work. The complexities of questions in biomedical sciences necessitates that we bring together physicists, biologists, chemists, computer scientists, and engineers to focus on life sciences. The merging of these areas now define life sciences in the 21st century. This is one take-home message you will certainly see tonight. And what is the role of basic science research when our goal is to create the kinds of tools that are used to make diagnoses and discoveries that will completely change our understanding of biological systems? This evening, we're going to hear from three really fantastic scientists who are making this leap by developing new microscope technologies that will help researchers around the world do neuroscience, genetics, just about every field of biology. The work this group is doing is astounding, but you'll need to adjust your thinking when we're talking about their technology. We're not talking about the microscopes of Leeuwenhoek, or the microscopes that you looked and fiddled with the focus with an objective lens, but technology that is pushing the limits of known physics that requires the processing of vast amounts of data on the terabyte and petabyte scale. And that allows us to record real-time videos of living cells. It really is frontier science. Our moderator this evening is Oskar Hallatschek, the McAdams Professor in the Department of Physics and Integrative Biology. He leads a lab that is trying to understand how collective patterns of self-organization, emerge from the joint actions of heterogeneous parts or individuals, organisms. The phenomena studied include evolutionary adaptation, random genetic drift, epigenetic, epidemic spreading, collective motion, synchronization, and jamming. Although these phenomena occur in many complex systems, his experimental efforts are focused primarily on microbial systems that he can study in his UC Berkeley lab. Okay, with those words, over to you, Oskar.
Thank you, Mike. It really is a great pleasure for me to play a role in this event about what really feels like a revolution in imaging. I myself am not someone who is pushing the frontiers of imaging. Instead, I'm a user. I'm constantly colliding with those frontiers in my everyday research. What keeps me up at night is trying to understand how bacteria make it possible to rapidly adapt to almost any kind of stress they encounter, how they rapidly adapt to antibiotics, which we try to use to attack them. Now, in the last 10 years, we have learned a lot about how and why microbes evolve very rapidly. But we know this mostly in our lab conditions, where we put microbes into little test tubes and shake those test tubes really well so that there is no spatial structure in there. But bacteria in the wild, they often live in communities where they have neighbors and they talk to their neighbors and they cooperate with their neighbors to build elaborate polymeric substance, polymeric structures, and virtually what structures that feel like little cities of microbes. However, we don't understand really how evolution works in this context. And part of the reason we know so little is that we can't watch them grow, divide, mutate, and evolve. So I believe if we could just look a little bit closer at these structures, and for longer times, we would be able to learn so much more about evolution of these critters, which is one of the reasons why I'm really excited today to hear from my three marvelous colleagues about how they push the frontiers in imaging and make my life to work in this field of evolution easier, but also of many other biologists working in lots of different fields. So I will now ask Tij, Na, and Gokul to share a bit about their current research and their vision for the future. But let me stress again that please don't hesitate asking any questions. We have substantial time to take any question and make this a lively discussion, which should be as interactive as somehow possible. So on to our first speaker, that's Robert Tjian. Bob is a long-term professor in the Department of Molecular and Cellular Biology. He joined the Berkeley faculty in 1979. He was trained as a biochemist at Cal and Harvard. He pioneered the discovery of human proteins called transcription factors that unlock the genetic information encoded in our chromosomes in the process called gene expression. For this work, he's received many awards, really too many to list here. But let me just mention he's an elected member of the National Academy of Sciences and an HHMI investigator since 1987. He also held several leadership roles, including, perhaps most notably, he was president of the Howard Hughes Medical Institute from 2009 to 2016, and currently holds the Li Ka-shing Chancellor's Chair in Biology and serves as scientific advisor to the Chan Zuckerberg Initiative and the Bio Hub. So, as I briefly mentioned, Tij's primary research interest has been for a long time to understand the machinery that is used to read off the information that's stored in our DNA, in the hereditary information, and turn this into proteins, the little machines that do most of the work in our cells. So Tij, could you tell us now a little bit how advanced imaging comes into play to learn more about this machinery?
Absolutely. Thank you, Oskar. Great to see you all. I'm the warm-up act for my two colleagues soon to come on and tell you about the exciting work that they've been doing. I'm from the undergraduate class of '71 here at Cal, working with Dan Koshland. And I'm very glad to tell you about kind of a little revolution that's happened quite late in my career. As you heard from Oskar, I was trained as a classical biochemist where we did experiments in test tubes, not in living systems so much. But about 10, 15 years ago, I completely switched my lab to using microscopes and to look at the biological process as you like, in vivo, in living cells. So I'm now going to share my screen with you and show you just a few slides of what I find most exciting about what we're doing right now. So hopefully you'll be able to see my screen. Everybody can see that? Great. So why am I showing you a galloping horse? Well, the point here is that for the last 150 years or so in biology, we've quite often been limited in our ability to actually watch living systems working in real time, and particularly at the minute nanoscale of protein molecules. You know, obviously with electron microscopy and X-ray crystallography, we could see molecules, but they were dead molecules, usually either frozen or fixed. The key here is a revolution which was really initiated by one of our current colleagues, Eric Betzik, in overcoming the diffraction limit of light microscopy. We were able to start to look at the 5 to 10 nanometer resolution and be able to see individual molecules. And so what I'm going to tell you about just in a few minutes is this revolution in what I call super resolution light microscopy and ability to watch molecules in motion in real time in living cells. Now, what is that going to actually look like? And here's just a few examples of what the machinery looks like. What we're actually looking at here is a cell that we're sectioning. You can see that the microscope looks nothing like your classical lone hook microscope. And on the right, what you see is actually individual protein molecules that we have fluorescently labeled that we can follow the movement of. And by using computers to actually analyze the trajectory of these moving molecules and their speed, we can calculate all kinds of things about the way these molecules actually operate. This was absolutely revolutionary for us because, as I said, up till now, we could see molecules that were fixed, but never ones that were actually moving. So we can now see how fast they're moving and what kind of restrictions they undergo in the living cell. Here's a vivid example of a molecule, which is the Huntington protein, which causes the Huntington disease. And on the left in the green, you see these aggregates, which is characteristic of what happens in the brains of Huntington's patients, usually in mid or late life. But what's really interesting is the movies on the right, where you had one very large green blob with these purple fireflies running around it. And you'll see that the purple fireflies are getting caught in that green mesh. And it turns out that part of the reason Huntington's mutations cause disease is its ability to disrupt the process that Oskar briefly mentioned, which is gene regulation, the expression of proteins in the right place at the right time. And in this case, in the striatal neurons, where things have gone wrong. And part of it is because the machinery has been hijacked by these aggregates in the Huntington's brain. So that's just one example of something we never could have seen before. And we can actually measure many, many parameters of these proteins and how they operate with each other. Another consequence of this super resolution microscope is not only can we detect motion, but we can see what the disposition of molecules in space are in three-dimensional space, not two-dimensional space. And that was extremely important if you're looking, for example, in my case, what's happening inside the nucleus of a cell. And so this is just one example. And the last thing I want to tell you is that although, as you all know, Berkeley is really famous for its ability to make new discoveries in basic science, in this case, biology and molecular biology, but very often our discoveries will lead to better understandings of disease. And that's one of the reasons I showed you about how we can look at Huntington's in a completely different way. And so this last slide just illustrates the fact that this new technology of what we call single particle tracking ability, literally the ability to watch the movement of individual proteins, whether they're mutant or wild type, in other words, normal, we can then discover new biology that could, when something goes wrong, how the behavior of these molecules changes. This has allowed us to develop new high throughput robotic systems and computational tools to screen for drugs that we never could screen before. And most importantly, as some of you may be aware of, even though there are thousands of drugs out there on the market, we've actually only targeted a very small fraction of the total number of genes and genetic elements and proteins that cause disease. And so the blue and green pieces of pie here are the small fragment of the entire proteome that we should be able to drug. And so one of the greatest aspirations we have of the technology I just showed you is not only to discover new biology, but to actually use it to expand the druggable proteome and get much better disease control. And with that, I'd like to thank you and be very happy to take questions.
Okay, thanks very much, Teach. It's really amazing to see how basic science can lead into actual real world application and even the development of new drugs. What struck me about the pie chart that he showed in the end was that just a tiny fraction of the proteins that actually are in our cells can actually be targeted by drug. And there's this large dark matter of undruggable proteins at this point. Can you tell me a bit, how this comes, why there is such a large undruggable portion of that?
Yeah, that's
why, and why imaging could help to actually find drugs that attack them?
Yeah, so there are a lot of reasons why the druggable proteome has been so limited. Traditionally, there's been several modalities of drugs, small molecules like a pill that you take, an antibody, or a biologics like insulin. And these things all interfere with the target molecules in very specific ways. And if it doesn't work that way, we can't drug the molecule. What I think the imaging technologies and genomics technologies, which I didn't discuss, will allow us to do is to reveal many more drug disease-oriented targets that we had no access to. And that's partly because I think that the imaging allows us to actually watch the target molecule in the disease state, which is something we never could do before. And so we had no way of actually knowing when something was going wrong and why it was going wrong. And today we can actually do that. And so we can develop screens that we never would have imagined doing just a year or two ago.
Fantastic. So, the way, let me understand a bit better how this drug discovery would work. So would it work? I mean, you had this one slide with the fireflies that you could watch very carefully. And then it seems like the key insight came from realizing that these fireflies were actually interacting with this big blob and therefore changing the way they're moving. So is this what you envision, that you can also now systematically look at other proteins, how they move, whether it's just regular diffusion or when you add some chemicals, whether this will change in some significant ways? And if it does, then you would say, this chemical interacts. Is that the way you envision it?
That's exactly correct. So in the example that I showed you, where you have the Huntington's green aggregate and you have the purple firefly flying around and then getting stuck, the thing that we would be screening for would be small molecules that would actually unstick the purple molecules. And we would never been able to measure that in the way that we measure it now. And in fact, we're actually going to probably do that in a startup that has been formed.
Just out of curiosity, is that what we saw there? Is that regular diffusion, for which you don't need any biology because thermal kicks are moving the molecules around, or do you expect also the cells are burning energy in order to live? So is there something more funky going on?
There's definitely many interesting things that we did not expect from the diffusion properties of proteins in living cells. So if you look at that same purple protein in a test tube in aqueous solution with nothing else around, it diffuses by Brownian motion just like you would expect. Mm-hmm. But in the living nucleus of that striatal cell, its behavior is anything but Brownian. It is highly anisotropic, which means it's very directed, almost as if it's limited in its movement, where it can go and where it can't go. The way it explores its space, the volume that it's sitting in, is not at all what we would have expected from the test tube experiment. And that's critical question that we're now being able to address. So
it's moving slower or faster? Because that's...
it's not a question of moving slower or faster, but moving in a way where it tends to go back to where it came from. In other words, it's not random. It's not as random as we thought.
That's interesting.
Exactly what's creating this anisotropic movement is still not clear. But we suspect it's because, remember, the nucleus is full of many other proteins, hundreds of thousands of other proteins, which this purple protein is going to interact with, perhaps not as effectively as it does with the big green blob, but it does with the others. And that will influence its movement.
Mm-hmm. Really interesting. And maybe stepping back a bit, just about your own, from the bird's eye view on your own research. You said you were trained, as a traditional biochemist, and then you pivoted your lab to become a very different lab, and actually doing frontier science and imaging. how did you make that possible? I mean, where's the flexibility in the system to
I think that that's one of the great advantages of being here in Berkeley. Without the graduate students and postdocs, and in my case, young colleagues like Gokul and Na, who you will hear from next, it would have been very difficult for me to go and switch myself from a classical, what I call bucket biochemist, to a high resolution imaging guy. And to do it in very complex biological systems like the brain. And so it is in fact the human resource of Berkeley that allows me to do this. And as you notice, it requires not just biologists and microscopists and engineers, but software, you know, computer science, physics, optics, all of these things have to come together. And I think it would have been virtually impossible for me to do that in almost anywhere else.
Okay, thank you, Teach. Thanks very much for sharing your research with us. I think this is a great time to move on to one of your younger colleagues, Gokul, and to transition from actually looking at individual molecules, but to entire groups of cells, to tissues, even to whole organisms. So the colleague that I want to introduce is Professor Gokul Upadhyayula. Gokul is an assistant professor in the Department of Molecular and Cell Biology. He's interested in bioimaging, microscopy, multimodal imaging systems, and artificial intelligence used to actually enhance imaging. His research group at the new Advanced Bioimaging Center uses cutting-edge pre-commercial imaging technologies such as the next generation adaptive optical multifunctional lattice light sheet microscope. That's a really wondrous instrument I think we will hear about, and it enables imaging across scales spanning several orders of magnitude in space and time in specimens up to several millimeters in extent. So please, Gokul, please tell us a bit about your research and the projects you're currently working on. Please.
Oskar, thank you so much for the kind introduction. And it's really my pleasure to be here to tell everybody what we're up to. So right now I'm a scientific director of the Advanced Bioimaging Center, where we are building these pre-commercial instruments, specifically focusing on capabilities that are not available commercially. And the idea is to make these technologies accessible to scientists from around the world, and have them basically give us a benchmark in terms of what we need to be working on next. So today I'm going to be focusing a little bit about the journey that kind of went through in order to get to where we are today, starting with my childhood obsession and actually continued passion in computer science, which took a sharp turn to specializing in engineering during my graduate work for my PhD. And the biggest transition to actually studying biology, to specifically cell biology during my postdoctoral work at Harvard. And it's a combination of these three areas, the computing, the engineering, and the biology, that has basically led me to work on advanced microscopy, because it really is an integrated and innovative field that's evolving at more or less breakneck speeds. So without further ado, I'm just going to go ahead and share my screen. So in our labs, what we want to do is we want to watch cells, which are the basic fundamental unit of life, in action to understand how life works. So compared to an ant, human cells are more than 100 million times smaller in volume. So do you see that cell that I put right next to this ant? It's like right there. To be able to see that, we need to make the invisible visible. And we do this by building microscopes. So the microscopes that we build look a little bit more complex than what we're used to from our high school microscopes. And they look something like this. And it helps that I'm part of a paparazzi of sorts. So instead of taking pictures of people, I'm more interested in taking pictures of cells using modern microscopes. And by using these modern tools, we've been able to observe, analyze, and learn where specific cellular compartments are located, how they're organized, and how their behavior changes with time. And the study of these types of dynamics up until a few years ago has been mainly limited to the cover slip and in 2 dimensions. So this is basically where we come in, a team of microscope developers, software engineers, biologists, chemists, working together to really push things forward. So what's really different about these modern microscopes? Well, let's take this cell on a piece of glass as an example to compare the widely used confocal microscope for 3D imaging. So the confocal microscopy has been around for a while, and almost every cell biology department has at least one of these microscopes. So do you see the glowing spots on the surface of the cell right here? So these spots, when I play the movie, they'll appear out of nowhere because they're building this molecular basket to be able to bring food into the cells. And sometimes viruses hijack this machinery in order to gain access into the cells. So either way, we can watch basically the dynamics of these cells in 3 dimensions to understand how cells eat. So when I play this movie, you can see the comparison between what is the gold standard confocal microscopy compared to the modern microscopy that we're using in the lab called the lattice light sheet microscopy. With the lattice light sheet microscopy, we are basically shining a super thin sheet of light to minimize the risk of damage to cells. And because we have that really thin sheet of light, we have amazing contrast that lets us see what's going on inside the cell without really stressing them out. Using this form of microscopy, we can study how viruses get in and infect the cells. So in this example, we watched a cell interact with rotavirus particles. So this particular type of virus is very highly contagious, which causes over 200,000 deaths worldwide each year. By watching these viral particles interact in this movie with the cell, we can observe how this virus interacts with the cell. We can watch how the viral particle moves on the surface of the cell and inside the cell. And of the hundreds of viral particles that we've tracked in 3 dimensions, we're able to, we discovered that only a fraction of them actually make it into the cell. And of those that actually make it into the cell, most of them are degraded by cells in these specialized recycling compartments. Only a few viruses escape this fate, and that lets them replicate and spread. And by studying how these viral particles get in, we're able to identify the interacting partners of these viral particles, like at the molecular scale, to understand the rules that these viruses play by. And only by understanding these rules, we can start to outsmart them by surgically targeting the mechanisms that allowed the viral entry to take place in the first place. This type of 3D molecular imaging only became possible in the past few years. While we can watch isolated cells on a piece of glass at a great level of detail, that's not really their physiological environment. Cells didn't really evolve on a piece of glass or in isolation. And to study their natural behavior with just as much detail in a physiological environment, we need to build new microscopes, such as combining our lattice light sheet microscopy with adaptive optics. And you'll hear a lot more about adaptive optics from my colleague Naji in the next talk. So using adaptive optics and the lattice light sheet microscope, we can unscramble the light and deblur the images that are coming out of the microscope in order to be able to see the cells and the subcellular dynamics in living organisms or transparent organisms like the zebrafish very clearly. And in these systems, we can learn how cells organize themselves to form tissues and organs in 3 dimensions over the course of days. And by watching the interaction of hundreds of cells simultaneously, we can really understand how they communicate with each other, how they coordinate various actions and tasks with each other. To demonstrate the promise that this technology holds, we've partnered with some of the best scientists from around the world to investigate a range of basic science questions. In this experiment, we watched an area near the zebrafish ear to study how immune cells patrol to defend us from infections, and to see what these cells actually even look like. In this movie, you can see there's 2 types of cells, the immune cells that are crawling around. One that's moving very rapidly and very aggressively. That's going to pop up in just a second here. And then another immune cell that comes from a different region. It's more calm, kind of moving and surveying. So you can see that these cells are constantly moving to figure out where they're needed. And they're constantly sensing their environment for chemical cues to understand and to know whether there's stranger danger. In another example, we studied how cancer cells metastasize. So we did this by injecting human breast cancer cells into zebrafish. And the green are cells that are labeled, or fluorescently labeled, are the human breast cancer cells. So in these examples, you can see that these cancer cells are using the blood vessels, which are shown in magenta here, they're using these blood vessels as their personal highways to move about the cabin, so to speak. So we see them rolling or squeezing through the blood vessels. And in the last example, we see what looks like Ridley Scott's trailer for the next Alien movie. The cancer cells are extravasating in an attempt to metastasize and spread through the body. And using these modern microscopes, we can watch how the cells really act and on their home turf. And in all these cases, we're able to see the dynamics inside these cells more clearly than ever before. But as with all new technologies and new capabilities, our new capabilities come with new challenges as well. For us, the big one is how do we manage the massive amount of data that's coming out of these microscopes? So we went from looking at the action inside of isolated cells in 2 dimensions to being able to looking at simultaneously the action of cells, hundreds of cells in 3 dimensions inside of living organisms with the same level of detail. So the data generation rate for us is insane. We generate anywhere from 1 to 3 terabytes of data per hour. And for our more experienced audience members out there, that's roughly equivalent to about 2 million floppy disks an hour. For the millennial audience members, that's roughly 500 DVDs an hour. And for the Gen Z, we're filling up about a dozen iPhones every hour. The data we collect is an embarrassment of riches. And now we want to know how to visualize this data, how to draw meaning from it, and how do we get this technology into the hands of scientists from all over the world. So to get there, we just have to collaborate even more. We'll have to find scientists who are developing biological tools and chemical tools. We'll have to work with physicists and engineers to build, operate, and maintain these advanced microscopes. And we'll have to join forces with the mathematicians and the computer scientists to get help in analyzing the immense amounts of data coming out of these microscopes. So we're building an imaging center to give free and open access to these advanced microscopes to scientists from around the world. And we want to use Berkeley as a hub and these advanced imaging as a hub to bring together and close the loop between the microscope builders, developers, the biologists, the data scientists, the computer scientists, and to work as a team so that we can solve problems together that none of us can individually. And thanks to these new imaging tools, the frontier of science is open. So let's take a look together.
Thanks a lot, Gokul. This is amazing. It really feels like magic, what you're doing. The movies give an amazing impression of what cells are doing over time. It feels like going to the movies. One almost forgets how much cutting edge technology and work is behind these instruments. So how long does it take to actually build these instruments? It seems like from your last movie, it seems like it takes a village to even build them.
Yeah, so to this microscope that I showed you in the last movie, that took about a team of about 7 folks, almost a year to design. And then there was enough interest and excitement around this instrument that we've built about 7 of them in parallel. So that means that every mistake that we made it 7 times and fixed it 7 times to start.
In Berkeley?
No, so we're 7 around the world to start. 2 at Janelia, 1 at Harvard, 1 at MIT, 2 at Berkeley. And to build the first prototype took us about 6 months or so, but we're getting faster and faster with every iteration that we're starting to build. So now we can more or less assemble the entire instrument in about a month or 2, and then another month or 2 to align and work through the software to understand and learn how to operate these instruments, because we want to drive them as Ferraris and not as Fiats. So,
yeah, what I find amazing is that you not only achieved this amazing feat to build these imaging Ferraris, but that you really can learn new biology with them. So you had these amazing movies. So maybe going back to that last cancer cell movie, how do you in the last state, it looked like the cell, or you said the cell is escaping now from the vessel. So how do you know that it's actually pushing out there and escaping from the vessel?
That's a great question. Let me share this one back up again. So we know that this particular cancer cell is extravasating because we have the level of detail in order to analyze the surface area of this particular cancer cell outside of the blood vessel versus inside the blood vessel. And what we notice is that the surface area of that cancer cell increases over a course of about an hour and a half by 50%, meaning that it's basically spilling its body outside of the blood vessel. And by doing that, so we know that it's actually coming out and not going in.
Mm-hmm. I see. Then maybe also about this other movie about these ghost-like immune cells that were floating around. There were these blue blobs, and can you maybe say a bit more about that?
Yeah, that's a great question. So we wanted to also get a sense for, we wanted to get a sense for how these immune cells were sensing their environment. And one of the best ways to understand that is to put in fluorescently labeled sugar. So in this case, we're using dextran. It's a fluorescently labeled sugar. And the idea is to understand how the cells are eating. So the way the cells are sensing their environment is by taking a little bit of their environment and kind of, internalizing it. So you can see that one of the immune cells, not this one, but the other one, has internalized a lot of its environment. And you can see that in its more or less stomach, so to speak, like these big endosomal globules. And then you can also see that there's about 11 different cell types in this field of view. You have skin cells, you have the brain cells on the right. All of them eat and internalize their environment for nutrients, but they all do it at different rates. So that's the reason we kind of injected labeled fluorescent sugar to be able to understand and track that particular behavior of eating.
What's also striking here, I feel like, is that these immune cells, they seem to have like almost different characters. Like one, as you said, is very aggressive, and the other one is like calmly patrolling, these cells. Like,
yeah,
how consistent are these different characters, and do you know what triggers that behavior?
Yeah, no, this is something that we're just starting to work with scientists to understand a little bit better, because again, this is probably one of the highest resolution dynamics of these cells. So this is basically just getting started, and it's very exciting.
Okay, amazing. Thanks a lot, Gokul. I think we should be moving on now to our final speaker of today. So let's move on to actually the most complex tissue in our bodies and animal bodies, which is the brain. And to this end, I want to introduce Naji. She's a colleague from the physics department. Naji is the Luis Alvarez Professor in the departments of physics and molecular and cell biology, and her research interests include neuroscience, microscopy techniques, in vivo imaging, biophotonics, biophysics, adaptive optics, and optics. Her team is developing exciting next generation optical microscopy methods for understanding the brain, while also applying the imaging technologies to understanding neural circuit computation in the visual pathways. So now, can you tell us now a bit about your research, and please share your screen.
Great. Thank you very much, Oskar, for the introduction. It's my great pleasure to be here and to be sharing with you how we can use optical physics to understand the brain. I am particularly excited today because the optical physics method that I will discuss just won 2 astronomers their Nobel Prize today. But before we get there, let's talk about our brain. Our brain is not very big. It's about 3 pounds in weight and about 1,000 cubic centimeters in volume. But it contains many, many specialized cells called neurons. These neurons communicate with each other through a special structure called synapses. Any moment, information flows through about 1 quadrillion synapses that are from 100 billion neurons. Together, neurons and their synapses serve as a biological basis of all our mental activity. Even for a much smaller brain, for example, the brain of a mouse, there are around 100 million neurons communicating through 100 billion synapses. To understand the brain, we need to know what is happening on the level of individual synapses. An ideal mind-reading machine, therefore, should be able to monitor the activity of neurons at sub-micron spatial resolution so that it can see individual synapses, but also ideally at millisecond scale time resolution so that it can keep up with the speed of information transfer in the brain. If we can express in the brain fluorescence protein whose brightness is correlated with the activity of neurons, we can indeed build such a mind-reading machine using concepts of optical physics. So my lab built microscopes that allow us to understand the brain at resolutions that will allow us to see individual synapses as well as follow the flow of information. So even with the best microscope that people can build, however, it is still very challenging to image the brain. Partly is because mouse brain, like the brain of all adult vertebrate animals, is opaque. This makes it very difficult for light to penetrate through it. With a conventional microscope, we cannot even see through the first 1 millimeter of the brain, which is called neocortex. This is a structure that allows us to have a conscious perception of the world. For example, this is an image of the brain at about 0.72 millimeter or 0.03 inch depths. Hardly anything can be seen here. This is because the light used to image this part of the brain got distorted by the brain itself. Now, a different branch of physics has been battling this problem for a very long time. To study distant stars, we would like to take a very sharp photo of them. However, before the starlight can reach the surface of the Earth and get connected and imaged with a telescope, it has to travel through atmosphere, where turbulence distorts its wavefront. As a result, when we look at the center of our galaxy, we would get a very fuzzy picture, making it impossible for us to study what is going on in there. But if we could measure how this light gets distorted, we can then correct this distortion and form a very sharp image. Over the years, astronomers developed just such a technology called adaptive optics. By removing the distorting effect of the atmosphere, it allows ground-based telescope to take extremely sharp pictures of distant stars. So here at the bottom, with the help of adaptive optics, astronomers can see clearly the stars that are orbiting the center of our galaxy, where there is a supermassive black hole. This exact work of using adaptive optics to study the center of our galaxy is just won 2 astronomers from Berkeley, and UCLA, the 2020 Nobel Prize in Physics that was just announced today. In my lab, we developed adaptive optical methods specifically for imaging the brain, where we face a different set of challenges from those in astronomy. Without going into detail of our method, the technology we developed allow us to image deep into the living brain, and we can go from seeing almost nothing to be able to see all the synapses on this neuron. By being able to see deeper, we were able to make new discoveries. One research direction in my lab is to understand how our brain processes visual information. When you look at your monitor right now, image was first formed on your retina. From there, visual information goes to a structure called LGN, which is a part of the thalamus of the brain. Thalamus then sends this visual information to the primary visual cortex, which further processes information and gives you the sensation of seeing. Visual cortex has a layered structure. Across all the different layers, neurons are found to selectively activated by different types of visual features. For example, if we present a mouse with images of a grating moving along different orientation, along different direction, having different orientation, some neurons will only become activated when the grating is along their favorite orientation. With our microscopy measurement, we will see that those neurons' fluorescence brightness will increase in response to the orientation that they like. We call this kind of neuron orientation selective neuron. Some orientation selective neurons also become active when the grating is drifting along only particular direction. So we call this particular type of neuron direction selective neuron. Such orientation and direction selectivity on the level of single neurons in the primary visual cortex are considered to underlie how we process visual information, such as orientation or motion. As you can see, orientation alone from the picture on the bottom left-hand side, orientation alone provides enough information for us to recognize this famous painting. Similarly, we are very sensitive to any motion in our environment. If you're meeting friends for a movie, you can easily find them if they wave at you. For many years, people believe that this orientation and direction selectivity is the only property that neurons in the primary visual cortex possess, and that the neurons in the thalamus do not have this property. But to know whether this is actually true, we really need to be able to image the synapses from the thalamic neuron in the visual cortex directly. To characterize the property of those thalamic inputs into the primary visual cortex, we can transfect those thalamic neurons with a fluorescent activity sensor. And then we can image their projection into the visual cortex. With the help of adaptive optics, we were able to get very sharp images of those synapses, hundreds of microns into the brain. We then present the mouse with gratings of various orientation, and those gratings are also moving towards different direction. You can see that their synapses will become brighter when their favorite grating is being presented. And for many of those synapses, they do not have a favorite orientation. So this is what people has believed to be true for 70 years. But to our surprise, we also found about half of the synapses to have orientation selective as well as direction selective properties. Interestingly, our ability to detect such selectivity depends on using adaptive optics. Without adaptive optics, many synapses would appear to us to not to select for any orientation or direction. This can actually be very easily understood. Imagine that you want to know the favorite food from a group of friends. If whenever you ask the question to one friend, all other nearby friends also shout their favorite food, you won't be able to find your answer. Without adaptive optics, similarly, the response we get from a synapse is contaminated by all the nearby synapses. Just like adaptive optics can help us to resolve stars orbiting the center of our galaxy, adaptive optics also allow us to focus on the synapse of interest alone and get this response. In this case, by using adaptive optics and applying for brain imaging, it allows us to discover something that we did not know about the visual system before, that thalamic input into the primary visual cortex are already orientation and direction selective. So here is just a one example of how optical physics concept, such as adaptive optics, can help us. Adaptive optics has been applied to astronomers for around 30 years, and great discoveries has been made. And we hope by applying adaptive optics to microscopy, we will be able to similarly make a discovery in biology, and in my particular case, to be able to see the brain in a new light. Thank you very much for your attention.
Thank you, Na. It's amazing how you transferred this technology from astronomy into biology, and now it allows you to look into the brains of mice. As you mentioned, today is a very exciting day for Berkeley and the UC system as a whole, because a Nobel Prize, 2 Nobel Prizes were awarded in physics, one for Reinhard Genzel in Berkeley, and second one for Andrea Ghez from UCLA for discovering this supermassive black hole in the center of our galaxy. Can you tell us maybe a bit more how actually adaptive optics was significant for their work? So that you better understand the Nobel Prize?
I knew you would ask this question. I'm very excited to be talking about it. So this is today's announcement from the Nobel Committee that the half was awarded to a theorist for the discovery that black hole formation is a robust prediction of the general theory of relativity. And the other half was rewarded to Reinhard Genzel from Berkeley and Andrea Ghez for the discovery of a supermassive compact object, in this case, a supermassive black hole at the center of our galaxy. And adaptive optics was employed by both astronomers in order to make this discovery. And this is the same data that I was showing you before, but except now it's in a movie form. So this is an image of the star at the center of our galaxy. And without adaptive optics, as I mentioned, you have this extremely fuzzy image that wouldn't allow you to be able to see the individual stars. Mm-hmm. But with adaptive optics, your image can become so sharp that you can not only see the stars, you're also being able to track the orbiting of those stars surround this supermassive black hole. And by monitoring how those stars are orbiting this black hole, you can test general relativity prediction as well as get information about the mass of the supermassive black hole, just as one example. So this is really the most understood supermassive black hole in our whole universe, thanks to adaptive optics.
Can you actually step back a little bit? And at this point, I also want to pick up a question actually from the chat. Is there a really simple way to understand how adaptive optics works?
Yeah, so this is how, can you guys still see my screen?
Not anymore.
Oh,
no.
You cannot see my screen? Oh, sorry. Ooh. I'm sorry, I guess so you didn't actually see the, the nice video data of the, let's see, that. Sorry, I should have shared. I thought you were able to see it, but this is basically the data of the star, image of the stars without and with adaptive optics. And this is how they were monitoring the orbiting of the star over multiple years in order to study the supermassive black hole. Now, how does the astronomers do adaptive optics? It's actually fairly simple. So as I mentioned that the light coming from the star get distorted by the atmosphere. And so as a result, this distortion causes us to be unable to focus this light sharply and form an image of the star. But if you can reflect this light off a mirror, an adaptive mirror whose surface shape we can control, and then you can split off some of those starlight and then directly measure how distorted its wavefront was. And then you can send this information through a control system to control the surface shape of this deformable mirror, so that after the starlight reflect off the mirror, now you will have this perfect undistorted wavefront, which will give you a very sharp image of the star. So this is basically how astronomers use adaptive optics to get a very clear image of the stars. Now, when we apply adaptive optics to microscopy, we could directly use very similar method where we measure how the light coming out from a sample is being distorted and then corrected before we take an image of this sample. So in Gokul's talk, he showed you many beautiful images of how adaptive optics can help to improve the image quality. And in his example, his biological sample are transparent. For example, this is a zebrafish larvae. You can clearly see that you can easily see through the body. So the same kind of method can be applied in astronomy, from the astronomy adaptive optics to microscopy for transparent samples. Now for brain, which as I mentioned, is really opaque. So that poses a new set of challenge because we cannot directly measure the wavefront, just like you cannot really take pictures of stars if you have very heavy clouds. So in my lab, we have to really use, based on other physics concept, try to measure how those wavefronts are being distorted. So it's a little bit, we have to use different type of method to correct those aberrations.
Thanks, Nal. Maybe let me take another question here from the chat. This question was, I think, asked to Gokul, and I'm sorry I missed it, but I think the question actually applies to both of you. Thomas Klein here writes, it would seem that the main current limitation in this technology, imaging technology, is the sensitivity of the detector. Is there any hope of getting much more sensitivity there, or are you at the limit? Question to both of you, Na and Gokul.
Gokul, you want to go ahead?
Sure, I can start. So that's a great question. Yeah, so absolutely. The limitation at this point in terms of how fast we're able to image really comes down to the sensitivity of the camera and how fast we're able to convert the signals that the camera is recording into data that can be transferred to the hard drive. That being said, we can now balance these limitations that exist by kind of compensating with sample prep technology. So by using brighter fluorescent reporters instead of proteins. We can use chemical dyes. That greatly helps. And we can optimize it such that we can address a specific biological question with single molecule sensitivity if that particular question can benefit from that. And I know there's a lot more work, and we're working with camera developers now in order to understand basically their roadmaps in terms of what is the current limitations that we as scientists are facing from the samples that we get, in order to understand how long it will take, both in terms of the engineering effort to overcome that. Now, maybe you can add some.
So as Gokul said, the sensors development, I would think that we're actually very close to what is physically possible. We're at almost 100% quantum yield, which means we can detect almost every photon that is hit our sensor. But so what we can really help is really also pushing on the labeling technology, the fluorescent molecules. The brighter they are, then the more information we can extract from the sample. So that requires the collaboration and work from chemists and protein engineers to give us better and brighter sensors. Okay,
maybe a final question also from Thomas Klein. He writes, this is about adaptive optics. He writes, I'm puzzled. I thought a large part of the problem was intensity, loss of light as you go. How does adaptive optics overcome the absorption problem?
Yeah, so adaptive optics cannot overcome the absorption problem. There's many problems in opaque sample. So there's this light loss due to absorption, but typically we use the light we use to image them falls within the band where it's not being strongly absorbed by the sample. But another problem we have is scattering. Just like, you cannot see through my fingertip. So because most of the light are scattered backward. Scattering can also not be directly solved by adaptive optics. Our trick there is try to use longer wavelength light, which is less scattered. So the astronomers, when they look at the center of our universe, there's actually lots of cloud dust before the light from the center of the universe can reach Earth. So they were using 2.2 micron, which is in the infrared. The reason is that this longer wavelength light is less scattered by those dusts. As a result, we can collect light with telescope from the center of the universe. So similarly, we can use longer wavelength fluorescent molecules, which will reduce the problem of scattering.
Okay, thanks to all the speakers today, Na, Gokul, and Teach. We are now closing the session. Before we close, of course, or to close, I'm going to hand over the floor to Frances Hellman, Dean of the Division of Mathematical and Physical Sciences and President-elect of the American Physical Society. Frances is a member of the American Academy of Arts and Sciences. And for me personally, Frances is a fantastic colleague and mentor in the physics department. She studies condensed matter, specifically the thermodynamic properties of novel solid materials, especially thin film, semiconducting, superconducting, and magnetic materials. Her work has recently led her in unexpected but exciting directions. She's currently a member of the LIGO team working on the next generation gravitational wave detector. Frances' materials could end up actually as coatings for the giant mirrors in this detector because of the unusual properties of these materials. So please, Frances, take it away from here.
Well, thank you, Oskar, and thank you, Tej, thank you, Na, thank you, Gokul, for sharing with us your work and a glimpse into this next frontier of imaging. That was fantastic. I am unmuted. I'm getting notices about unmuting, but I'm pretty sure I'm already unmuted. Anyway, so yes, one of the best parts about being dean is getting to hear our faculty describe their research and convey their own excitement about it. It's completely contagious, and it's a great reminder of the reason we're all at Berkeley, just the excellence of the scientists and the excitement of the work they're doing. It has been a particularly exciting and bordering on exhausting day for Berkeley and for the University of California. The Nobel Prize is, that's always it's just an exciting moment for everybody, of course. And, you know, one of the interesting things is just, I guess it's always striking to see the crossover between different fields of science. You know, that adaptive optics, you know, designed for one field of science is so important to another. And that's another reason why people work at Berkeley, is just the ability to hear these breakthroughs in one field that can spill into another one so easily. You know, many of you know Reinhard Genzel was a postdoc with Charlie Towns. And so, you know, unfortunately, Charlie died a few years ago, because, you know, probably one of the greatest joys one could ever have in science, well, perhaps winning the Nobel Prize yourself is a pretty darn great joy. But, you know, having one of your students or postdocs winning a Nobel Prize would, without question, be an inspiring moment. And, you know, one of the points of this whole series of talks that we've been doing is how basic science just underpins so much that ends up being great and important technology or biomedicine type discoveries, but underpinning them is this really basic science. And of course, Charlie Towns was known for his discovery of the maser and the laser. And now we see lasers being used by scientists like Na and so forth. It's just it's inspiring how everything overlaps, everything comes together. And, you know, it's through tools like these that we've been able to peer into the center of the Milky Way and into the center of these cells that we were seeing. You know, I'm sure I can only imagine that you were as inspired by those images as I was, because they're fantastic. Those images are just breathtaking. So, I do want to say a special thank you to our alumni and friends for gathering and talking about the importance of basic science. This whole series is intended to shine a light on this type of science and to convey its importance. I also feel I'm reminded of another great scientist who also died some years ago, but on whose shoulders tonight's presenters also stand, and that's Erwin Hahn. Erwin was renowned for his work on nuclear magnetic resonance, and most notably for his creation of what's called pulse magnetic resonance sequences and discovery of the spin echo effect, which led to magnetic resonance imaging, or MRI as we know it today. I was lucky enough to know Erwin before he died. I think some of the others on this call also were. You know, and his foundational curiosity-driven research led to that he had no motivation to study biology per se, but his work on NMR, which turned into MRI, and how, you know, really led to one of the most important imaging tools we have today. So, you know, I have no doubt that the science that you've seen tonight from Tij, from Gokul, from Na, like Erwin Hahn's, will lead us to great breakthroughs in understanding the brain, disease, and other biological challenges. And that is the Berkeley way. These foundational sciences lead to breakthroughs that have technological and biomedical importance. Dean Bochin and I feel very strongly that while it's essential to look for cures and answers to important technology questions, we must also focus on the foundational work that underpins those cures and answers. It's always been one of Berkeley's greatest strengths and has to continue to be so. So, what are you all doing here? Well, your support and your advocacy for Cal and your interest and your love of the science just means everything to us and to the faculty like Tij and Na and Gokul and Oskar, and, you know, and to their incredible students. So we are incredibly gratified for your continued interest in Berkeley, which is of course alma mater to so many of you, and to the path-breaking work that's getting done here. So if there's anything you want to learn more about, or if you'd like to support any of the work that you've heard of or other work that you might know of, and there is deep need, and please be in touch with us. We absolutely want you to be part of advancing basic science and discovery at Berkeley. And so with that, fiat lux. I hope everybody's well and stays well. I'm not sure if I was supposed to see if there's more questions. I'm seeing lots of nice comments. Thank you for explaining. I want to note these before we sign off, because I think these are directed at our speakers. Thank you so much for explaining in a way that non-physicists can understand it. Fascinating, if a bit mind-blowing. There you go. So many, many thanks again to all of the speakers and the moderators, and fiat lux. Have a great evening.